So today I want to talk to you about enzymes and how they're critically important pieces of cellular machinery. But first, let's review the idea that biochemical reactions happen in the body all the time. Almost every cellular process involves a biochemical reaction at one point or another. The TCA cycle is actually just a series of different biochemical reactions in carbon metabolism. DNA replication, which needs to happen before a cell can go through mitosis, is also just a series of reactions. And this also applies to the expression of genes, going from DNA to RNA to protein. And we need enzymes because enzymes make all of these reactions go much faster. And let's look at this idea a little more deeply and how a reaction will go on differently when it has an enzyme versus not having one at all. So you may be familiar with the reaction where water and carbon dioxide can combine to form carbonic acid. And this is a reversible reaction, so it can go backwards and forwards. Now, when people make soda or any carbonated beverage, they'll start by pumping that soda can full of CO2. And while some of that CO2 will dissolve in the water in the can, the soda making companies are able to get a lot more CO2 in the water by using this reaction. The abundant CO2 will react with the water to form some carbonic acid in the can. And when you go to open the can, you'll hear a pop sound, which is really just a bunch of CO2 escaping. But after that, the soda will start to fizz really slowly. And what's happening here is the carbonic acid that was made before is slowly dissociating back to carbon dioxide and water as CO2 escapes. And that extra CO2 that's being made will come out of the soda solution, and you'll see it as little bubbles floating around. But what happens if you then take this person over here and he'll pick up a can of soda and take a drink? That person might notice the soda will start fizzing a lot more once it hits his or her tongue. And this is because humans have an enzyme in their blood and saliva called carbonic anhydrase. And this makes the carbonic acid turn into carbon dioxide and water much more quickly. So more CO2 will come out of the can and it'll fizz more. And this is just one of the many examples of how enzymes make reactions go faster. So how exactly do the enzymes make the reactions go faster, though? Well, they use a bunch of different catalytic strategies to push reactions along a little more quickly. And I'm going to talk about a few of those strategies just to give you an idea of what enzymes are doing. So first I'll mention acid-base catalysis, which happens when enzymes act like either acids or bases. Now remember that acids and bases are proton donors and acceptors. And if you look at this type of a reaction, which if you remember from organic chemistry is a keto-enol tautomerization reaction, we have a proton moving from a carbon atom to an oxygen atom. And since acids and bases are pretty good proton carriers, they could both help with this reaction and make it go a little more quickly by helping to move that proton around, instead of this molecule doing it by itself. Our next catalytic strategy is covalent catalysis, which happens when enzymes form a covalent bond with another molecule, usually their target molecule. Remember that covalent bonds involve two molecules sharing electrons. And looking at this reaction here, we have a decarboxylation reaction going on, which if you remember from organic chemistry is when a carboxy or CO2 group is being taken off a molecule. And if you remember, these reactions usually have a lot of electrons moving around. So if we had a covalently bound enzyme that could hold on to some electrons and be an electron carrier, or what some people like to call an electron sink, then that would definitely help this type of reaction move a little more quickly. Next we have electrostatic catalysis. Now if you remember, DNA is a very negatively charged polymer because of all the negatively charged phosphate groups that we find in DNA. So if an enzyme had a metal cation on it, like magnesium, we could use it to stabilize the negative charge found in DNA and make it a little easier to work with. And DNA polymerase, which is the enzyme that allows DNA replication to occur, does exactly this. And in order for it to help with DNA replication, it needs to find a way to counteract all of the negative charge on DNA, and magnesium ions totally come in handy there. So the last catalytic strategy I want to mention is a little more general, and it has to do with proximity and orientation effects. Remember that in order for two molecules to react with each other, which is usually what enzymes help out with, they need to physically collide at some point. If we have molecule A and molecule B, they'll only react once they crash into each other. And a lot of enzymes are able to bring two molecules close together so that these types of collisions happen more often, making the two molecules react more quickly. Also remember that the orientation of the two colliding molecules in space is also really important. If molecule A and molecule B collide, but one of them is upside down or not in the correct position, then the collision may not result in a successful reaction. So enzymes also make sure that the two molecules will collide in the right orientation. And all of this increases the frequency of collision in general, but also helps to make sure that those collisions are successful and result in a reaction. So what did we learn? Well, first we learned that the role enzymes play is to make biochemical reactions happen more quickly. And the next thing we talked about were 4 of the many different catalytic strategies that enzymes can use. We talked about acid-base catalysis, which helps with proton transfer. We talked about covalent catalysis, which helps with electron transfer. We mentioned electrostatic catalysis, which deals with stabilizing charge. And finally, proximity and orientation effects, which increase the frequency of successful collisions between molecules that we want reacting together.